Hi, good morning, AP Biology. I'm just going to play this topic 6.2 question for you here, and in an attempt to make it whoops, I guess I can't float on top when I'm doing that, but I'm going to just narrate it for you because this woman's
Hi, everybody.
audio
Welcome to
is really
AP Daily.
terrible.
This is AP Biology.
So, this video is about RNA processing and transcription. So it's about how you make it and also what happens to it after it gets made. So I'm just going to forward. What will we learn? These are always important to look at. So the flow of genetic information, what is that? What is transcription? You kind of know that. What mechanisms are involved, so what enzymes have to happen, what parts, and what are the different types of RNA, and what does each do. This we reviewed last week. You have your DNA, which is transcribed to RNA, which is translated to proteins. So you got all your genetic information stored in DNA. That's what you got from your mom and your dad. And then RNA is a copy of DNA that is used to make proteins. So that's what RNA is. And finally, proteins are what we're really going after here. Proteins are the big thing that runs our body, and ribosomes are what make those. Ribosomes are also made of RNA. So here's a little diagram of the process of transcription. First, you have to unwind the DNA. Then this enzyme called RNA polymerase has to add RNA nucleotides, and you can see that's them being added there. That's what the RNA polymerase is for. And then the newly synthesized RNA strand is here, and you see how it matches up with one side of the DNA called the coding strand. And then there it goes off out into the cytoplasm to go to the ribosome, but that's not transcription. So now there is something here about a 3' to 5' direction, and basically that just means that that DNA you learned before is anti-parallel. So that's something where you can read about it in the book if you need to. But basically what it means is one strand goes one way and the other one has to go the other direction, kind of, because the 5' ends are at a different side. So the three types of RNA here, mRNA, tRNA, and rRNA. mRNA is the one we've been talking about that takes the information to the ribosome to make the protein. It's basically just a copy of the DNA. tRNA is the transfer molecule that has the anticodon, which is in the language of nucleic acids, and it also has the language of proteins there. So that's the one we're going to talk about when we're going to translation. And finally, rRNA, that makes up ribosomes. And I'm just going to fast forward through this. You can definitely, like I said, watch this if you don't know these things and take notes about them. But here's a practice question, and this is what's going to be on your assessment. So first of all, in the AP Biology test, these skill focuses are kind of cool because there's something that is going to be applied in many different cases. So in this case, you're going to have to look at a visual representation or a picture, right? And then you're going to have to explain relationships between different characteristics of biological concepts, processes, or models. So let's look at the question here. So the question is going to be the one that you have to answer for your assessment. And let me read it to you here. First of all, the visual representation is transcription. So you can tell that because you see how there's U's on that one strand? That is a clear indication that you're using or you're talking about RNA, right? Because there's no uracil or U in DNA. And you see the DNA strand there, and it's labeled strand X on the bottom. And then you see the RNA being made from it. So transcription is a process involved in gene expression, and it includes the use of enzymes. Figure 1 represents the transcription process. Which of the following statements best explains how RNA polymerase is involved? So, A. RNA polymerase synthesizes a new DNA strand in a 5' to 3' direction. B. RNA polymerase synthesizes mRNA by base pairing with the coding strand. C. RNA polymerase copies the gene by base pairing with the non-coding strand. And D. RNA polymerase copies the gene by synthesizing tRNA from the antisense strand. All right, so you're going to figure out what you want to do for the answer for that. And then the second part of the assignment, but you can definitely, I'm going to skip over where she gives the answer, but that just gives you motivation, motivation to go and watch the video if you don't know the answer to this one. Finally, what you have to take away from it, and I'm just going to play that. Genetic information flows from DNA to RNA to protein. Transcription uses RNA polymerase, and it uses the non-coding strand of DNA as a template to produce an mRNA molecule. RNA polymerase synthesizes mRNA in the 5' to 3' direction while reading DNA in the 3' to 5' direction. So that just talks about the directionality. And then there's the 3 types of RNA again. And then she says, thank you. She's from New York, and her mic was constantly like so close to her face that you could hear her breathing, and it was really annoying. So, all right, go off and watch that video on your own, or go to the assessment, answer the question, and there's a second part where you have to explain why you picked that answer. Just FYI, so you know that. All right, and there'll be more being posted. This shouldn't take you too long. Have a good Monday, you guys.